Translation. Translation can otherwise be called as protein synthesis. It is the decoding of the genetic code into the amino acid sequences. It is the second step in central dogma. It describes how the genetic code is converted into amino acid. There are three important steps in translation. First one is initiation. During initiation, the mRNA, tRNA and amino acid all come together within the ribosome. The initiation site is the start codon. The start codon marks the beginning of coding sequence of a gene. It specifies methionine that is brought by tRNA that bears the anticodon UAC for AUG according to the complementary base pairing rule. Requirements of initiation. The first one is charged tRNA. Every tRNA has an amino acid binding site opposite to its anticodon arm. The tRNA needs to be charged to start the process of translation. Charging of tRNA occurs when the amino acid specific to its anticodon attaches to its amino acid binding site. The amino acid is carried at the 3' hydroxyl end of the tRNA molecule. The charged tRNA is called amino-acyl tRNA. tRNA is charged by forming ester bond between the COOH group of the amino acid to the 2' or 3' OH group of the adenyl group at the end of the acceptor arm as shown in the following figure. The charging of tRNA is catalyzed by the enzyme amino-acyl tRNA synthetase. The next one is ribosomal subunits. Ribosome is a site where translation takes place. Both the small and large subunits assemble along with the mRNA and other initiation factors to start the process. This figure shows the three different sites in these ribosomal units. The next requirement is mRNA. In mRNA, there are start codon and stop codon. There might be many AUG in a single mRNA. The right AUG is picked up by the ribosome. The recognition mechanism is different in prokaryotes and eukaryotes. In prokaryotes, the recognition site is Shine-Dalgarno sequence, whereas in eukaryotes, it is the 5' G cap. The codons on mRNA are read in 5' to 3' direction and the anticodons on tRNA binds to the codons on mRNA in an antiparallel fashion. That is, its 5' base hydrogen binds to the mRNA's 3' base. The first two bases from the 5' to 3' direction are constrained bases in the codon. It is vital for amino acid insertion, whereas the third one at the 3' end is not necessarily be a perfect match for an amino acid to be added successfully to the chain. It is explained before in wobble concept. The next process is elongation. During the elongation step, amino acids are added to the carboxyl end of the previous amino acid. The polypeptide chain grows as the ribosome moves from the 5' end to the 3' end of the mRNA. tRNA with anticodon complementary to second mRNA codon binds to ribosomal A site with appropriate amino acid attached to it. A peptide bond is formed between the first and second amino acid in P site and A site respectively. The formation of peptide bond is catalyzed by the enzyme peptidyl transferase present in the ribosome. The energy required for this process is provided by the hydrolysis of GTP. Now, the first tRNA in P site leaves the ribosome as it is detached from the amino acid. As the ribosome moves along the mRNA, the tRNA with second amino acid moves to P site exposing the A site. The A site is now free for the amino-acyl tRNA corresponding the next codon to bind to it. Then, as before, a peptide bond is formed between the second and third amino acid and the ribosome moves along a single codon, leaving the A site free for the next anticodon. This process is repeated until the ribosomes encounter a stop codon. The next step is termination. When the A site reaches a stop codon, instead of tRNA, release factor proteins binds to the A site. There is no tRNA available with the anticodon for stop codons such as UAA, UAG and UGA. The binding of release factors causes the release of newly synthesized polypeptide and mRNA from the ribosome complex. Finally, the ribosomal subunits also get separated from each other.